Okay, so let us start our today's seminar, and I'm glad to introduce our today's two speakers. Oleksandr Frei, which was my former colleague, was in the Bayram group several years ago, but then he joined the University of Oslo, and his fellow Bayram Akdeniz, and they will give a joint talk about the application of variational Bayesian inference methods in genomic research. And I'm also glad that me and Dmitry Korpatov have assisted a bit in the work of our colleagues. So colleagues, please.
Thank you. So let me see if I can share the screen. Do you see the slides?
Yep.
Yes. Okay. So thank you so much for the opportunity to present the research. Again, it's actually the second time I got a chance to present to your group. And this time it's with my colleague Bayram. Both of us work at NORMENT. NORMENT is a Norwegian Center for Mental Disorders Research at the University of Oslo. And yeah, we are very excited to present this research and get your input. The previous presentation was about 3 years ago, so November 2019. And even though it was just one seminar, it really was super helpful. It directly led to at least 2 papers already on bioRxiv, medRxiv, and in some journals in revisions. So one of these papers is a focus of the talk today, and it's the one in the title, the FINEMAP-MiXeR variational Bayesian approach to genetic fine mapping. And the other one I'll also mention that very briefly. And as last time, I'm absolutely sure it will be super helpful for me and for Bayram. And we are not experts in Bayesian inference, but very eager to learn from you and as much as possible. And that's why we are here. But also, please help us to make sure it's useful for you. So please ask questions, especially when it comes to domain about genetics. We are very happy to try to address these questions. So just ask at any time if you have anything in mind. All questions are going to be really good, I'm sure. So that's a plan. I'll start with an introduction, going over what's called the Genome-wide association studies, or GWAS. And then we'll talk about simple additive genetic model. It's so simple, if you look at the formulas, that we're a bit embarrassed. So you're excused if you feel it's too simple. But believe me or not, despite simplistic math, more like school level math, some of it is fairly challenging, and it kept me motivated and interested for like 6 years that I'm doing statistical genetics. It's not that simple, despite the formulas might look turning it into practice and getting to the bottom of how genetics work in real life is pretty cool. So then we'll talk about some math behind the MiXeR prior, the spike and slab distribution for genetic effects. And after introducing this, I'll hand over the stage to Bayram, and Bayram will do a deep dive into the fine mapping, existing tools for fine mapping, and then how we use the Atom optimizer to minimize elbow evidence lower bound, and then some results from simulation and real data. I think, Bayram, your real data will be about height, right? So meaning the human height, how tall or short people are. Like I'm 182 centimeters tall, so that would be a little bit about why I ended up being that tall or short. So and then I'll continue as if time allows, right? I think that's more or less flexible agenda. By the way, how much do we have for the whole, like including discussions? 1.5 or 2 hours?
About 90 minutes.
Okay. We'll try to keep the time. So yeah, if time allows, we'll talk about some of the challenges in statistical genetics, maybe discuss other techniques for solving this MiXeR prior, including SuSiE, this new method from Michigan's group. And yeah, some discussions, conclusions. Okay, let's get started. So trying to frame GWAS from machine learning perspective, I need to tell that input is a genotype matrix having n subjects and m genetic SNPs or genetic features. And so SNPs or features are columns of this matrix, and subjects or individual like human beings are the rows of this matrix. And we are trying to predict either binary or a continuous variable, like height, or if it's binary case control, we say, right? It could be a diagnosis, status of a, like, is it a patient or is it a healthy person? And within patients, it also could be, let's say, a medication response, right? If we can, based on genetic data, conclude that some drugs are not worth a shot, right? So people shouldn't really struggle to even try those drugs and suffer from side effects. That would be a lot of help for the precision medicine. Yeah, but basically, this is a G matrix, and at this point, it's not really important what those SNPs are. I'll try to explain a bit more on the next slide, but whenever you hear this SNP or genetic variant, SNP stands for single nucleotide polymorphism, you can think of these vectors of zeros, 1 and 2s. Again, next slide, I'll explain why this is 0, 1, 2 features that we are talking about. But for you, it's a predictor that aims to explain the target class, which could be binary or 0, 1, binary or continuous. How do we know that these features are informative, right? I think it's an interesting topic. And we are a lucky domain in a way that we don't even need to measure these features in order to tell that they're informative. Because for more than a century, it's like there is a lot of genetic analysis of heredity, looking how these phenotypes or target variables are transmitted from parents to kids, right? And how they're similar or different within siblings in the same family, that we can conclude that these genetic variants, they are informative, in fact, right? So we all say that, oh, this is like a tall boy or girl that comes from a father or from a mother, or this is a smart boy or smart girl that's also like parents were smart. IQ is heritable, height is heritable, and unfortunately, a lot of human disorders are also heritable, that they run in families. And more formally, this is estimated with twin studies. I won't go into the detail, but it's basically you have monozygotic twins and dizygotic twins, so kind of identical twins and non-identical twins. And the identical twins are more similar to each other genetically, right? They are identical. And if they are also a lot more similar in terms of their phenotypes, we can make an inference about how much variance of the phenotype is explained by genes. But quite recently, we actually got access to genotyping or measuring all of these genetic variants, and it enabled a lot of new research like the one we are doing here. So another interesting aspect about this is that we are pretty sure that those SNPs are causal in a way. Like normally, if you have this type of machine learning problem, you can only say that, well, you can predict the phenotype from the features, but you don't know, maybe there is some kind of complex confounding correlation going on. It's with genes, things are different because like we are born with our genes and phenotypes came later in life, right? So we grow to a certain height, we get certain disorders. So because genes, SNPs are imprinted in our DNA from day 0, right? We know that by studying this, we can actually understand the mechanisms of the disorders. And that's why we are not only interested in this kind of just machine learning prediction perspective, using a black box, right, that will take features as input and produce some expected plus y as an estimate. We are interested in how these things work, right? And so a lot of this is about understanding this mapping from SNPs to phenotypes. Going a little bit towards this simple additive genetic model, an embarrassing thing I mentioned is that we're only looking at the first order rules. Of course, they are weighted. It's a weighted classifier at the end, but we are not considering second or third order. And partially this is because we don't have signal to deal with this despite really huge samples, partially because it works well with the first order and it presents enough challenges, but that's what we are basically dealing with. So this GWAS, Genome-wide association study, is basically a feature selection problem. We would like to understand which SNPs are involved, which SNPs are informative, because in total, haven't mentioned that yet, but in total, we have millions of those variants. So we need to understand which ones are actually have something to do with the genetic risk of certain disorders. And the final point on this slide is that the G matrix and the phenotype Y, they cannot be easily shared because this is individual level data. Nobody wants his or her DNA to be publicly available or the information about diagnosis to be publicly available. So keep this in mind, the G and Y, if I manage to keep notation throughout this presentation consistent, G and Y is difficult to get access from another group or another study and require like heavy application and secure servers to work with this. Okay, so the basic technologies that give us access to this data called the genotyping, and I think you're maybe more familiar with sequencing, right? There's like a way of actually getting a DVD with all the 3 billion base pairs as a sequence, which represents the entire DNA that the person has. So this sequence has A, T, C, G letters, A, T, C, and G, right? 4 letters. Yeah, so you can sequence a person, costs about $1,000 US, and yeah, kind of a lot of this would be non-informative because, of course, for us to be humans, there should be something in our DNA that makes us humans and not like chimpanzee or banana, right? But still, there is a lot of differences within the human population, and those differences tend to go to a certain locations in the DNA, which we call SNPs, right, or single nucleotide polymorphism. So on this slide, a very illustrative example of a given SNP having 3 alternatives for 3 different individuals, A, G, or T. The rest is identical, as you can see. So we are only interested in order to study variation in phenotypes like height. We only need to look at those SNPs that are different, that are polymorphic across individuals. And there, the genotyping comes to the rescue because it's a lot cheaper, maybe a factor of 100 cheaper, maybe $10 per person. And it gives access to around 500,000 of genetic variants. Not the 3 billion, but 500,000, but also in effective features. They're actually different between individuals. Short note, why it's 012 on the previous slide is because most SNPs are biallelic. And so it's not A, G, and T. It's just, let's say, A and G across different individuals. One of them will be chosen as a reference. And you're basically counting how many reference SNPs a person have, reference alleles, let's say G in a given locus. And the reason why it could be 2, because one allele will come from father, another from mother, right? So the 012 is a number of reference alleles in a given individual for a certain SNP. So now doing this feature selection, looking into genetics of complex traits, we are interested in complex traits mostly. Complex means that it's not a single variant that defines the disorder. It's many multiple common variants with very weak effects scattered throughout the whole genome, and we refer to it as complex or polygenic traits. And because there are so many of those variants that have contribution to the phenotype, and because those variants each of them have a tiny effect, we actually need to involve statistics. We cannot just do a feature selection and say this is involved, this is not. We have to attach a p-value, which would give us a probability of rejecting the null hypothesis that this variant has nothing to do with the phenotype. That's why it's called statistical genetics, the whole area. And let's say more specifically how we do it for a case control study would be to, so on this slide in the bottom, you have 2 variants, 2 SNPs. First one has G or A alleles, and the second one has T or C alleles. And it's quite apparent that controls for the first variant have a lot more G variants, while for cases it's 50-50. Right, it's 3 green ones and 3 blue ones. So you count of frequencies, basically the frequencies of these different genotypes at every site, at every SNP. And if there is a substantial difference in frequencies of those genotypes between cases and controls, with, for example, exact Fisher test with this contingency 2x3 tables, you can conclude with the hypergeometric distribution, you can conclude that this difference in frequencies is significant. School math still. But in practice with the logistic regression mainly because there is some other confounders we need to control for, like batch effects or population. It gets complicated, but the basic idea would be like this: a simple Fisher test for allele frequency differences. And that would be an example of a real GWAS result for schizophrenia, published 2014, I think. Now there is a later version from a psychiatric genomic consortium. They discovered, I think, 128 variants associated with schizophrenia, and these are some of them. So you have each line correspond to a specific SNP. It would have certain location in the genome, and the two reference alleles. The first one have T or C, right? And then the next two columns will tell you the frequencies in cases, frequencies in controls, and you see how little difference is there, right? It's not like 90% frequent in cases and only 10% in controls. It's something like 53% versus 52%. And the odds ratio, if you're familiar with this term, it's like how much more prevalent schizophrenia is among people having one allele versus the other, is something like in the order of 1.1 or 1.05. So this is kind of the effect sizes we are dealing with here. Good thing is you can download these results without any registration and do your own research on them, because note that this is no longer about individual people, right? It's about basically, this is about SNPs, right? About genetic variants, and this information can be easily shared. So typically we represent the thing via this Manhattan plot. This is called Manhattan plot. The vertical axis here shows minus decimal logarithm of the p-value. So it goes up to 30, and that means that, yes, we did get some p-values 10 to the minus 30. It's not 0.05, but 10 to the minus 30. Yeah, but the point here would be that so for example, this top hit that you see as the most strong hit for schizophrenia disorder is on chromosome 6 in a region called MHC, and it relates to immune system. So kind of interesting biologically, but at this point, note that it's not the only point that is associated in that region, right? So if I show you this, you can see my marker. There is a huge number of other genetic variants that are also associated with 10 to the minus 27 and 24 and 21, etc. So why is that? Is it like in that locus, every single variant actually contributes to the risk of schizophrenia? And to explain this, and it's really relevant to what Bayram is going to present in the next part with FINEMAP, we need to look at what's called, fancy term, linkage disequilibrium, but it's basically the correlation matrix among the genetic features. So here we don't have any phenotypes. So it's just we're inspecting the data from the G matrix itself. And we've picked a small region, which consists of around, what I can tell, maybe around 1,000 features, 1,000 SNPs on chromosome 21. And we did basically a pairwise correlation matrix, Pearson correlation coefficients between those 0, 1, 2 vectors of the 1,000 SNPs in that region. And they go in blocks. So red color means high correlation close to 1.0, and white regions, no correlation of around 0, just noise. So you see they come in blocks. And interestingly enough, this middle block directly corresponds to a gene called PIGP, a long unpronounceable name. Those SNPs are always like inherited together, because probably the biology doesn't want if this gene gets split in half and only half of it is inherited from father and the rest comes from mother. So those have to be, in order for this gene to work, they have to go together. And then the histogram on the right, it corresponds to the total amount of correlations that a given SNP is taking, right? So the sum of correlations in a given row of this matrix. I can put it this way. But not all genes are inherited kind of as a single unit. This would be an example of another gene that has a lot of breakpoints in the middle, and it's kind of the net result that a person gets is a combination of the sections from both parents. So yeah, basically this is a big challenge, because then when these genetic features are correlated, if one of them make some impact for the outcome phenotype, everything that correlates, all features that are correlates with this important feature would also be significant. And we need to inspect this in lots of detail. Like this local zoom is a tool to visualize, like drill down on a given spike in the Manhattan plot, right? It's shown here, there's a big spike here, and we are zooming on this spike with local zoom. So it is kind of what we're going to do with FineMap, trying to understand which SNPs in a given locus, given what we know about the correlation pattern of the SNPs, seems to actually make a contribution to the phenotype. So, and in order to do that, we actually need some formulas and notations, right? So, going very briefly through the. Have you had any questions for the first? This just kind of later in the introduction part.
No, I guess no, because we haven't heard about the mathematical model itself yet.
Yeah, that's true. I thought it's good to start with just the domain introduction because, yeah, anyway. So, yeah, mathematical model, we have the phenotype denoted Y. It runs from the K index, goes across individuals, right? In total N individuals on this data set. The G is the genotype matrix. 0, 1, 2, and the beta is the genetic effects, right? The coefficients of regression in this model. In a matrix form, it's on the right-hand side. So G and the outcome phenotype are known, and betas and this residual term E are unknown, and those we are trying to estimate or make inferences about. So of course, a naive implementation for this, once we've genotype sample and we have measured the phenotypes, would be to do an OLS, ordinary least squares. But this implementation works badly because this is an ill-conditioned problem with too much correlations. So the conditioning number for this G matrix is too high. It has a very high ratio between highest and lowest eigenvalue. And therefore, what is very common in this domain is to consider the features one by one and like a naive Bayes classifier and test for significance of this correlation between phenotype and a given feature at a time. And those are referred to as z-scores in GWAS. So GWAS is testing SNPs marginally rather than in a joint linear model. And this first formula here is what we refer to as a simple additive genetic model. So it's linear across features. What is very helpful for us is that you can do inferences about the additive, this beta coefficients through GWAS z-scores. So the theorem that I'm not going to prove here, but the theorem is that if the phenotype is related to betas through the genotype, like they look very much similar, but now instead of the design matrix in this system of linear equations goes from being n by m, n subjects and m variants, it goes to be diagonal with m by m matrix, banded as I show. So it's related to that correlation matrix that I shown on previous slide. And it more or less has the same information to do the inference about this beta effects from the GWAS association summary statistics, which, as I mentioned, you can download for free. They are not individual level, not sensitive.
Can you please mention one more time what is Z vector and how it's computed from Y and G?
Yes. So Z vector, the reason it's called Z is because it's a Z statistic. It's a Wald test. So ideally it should be called T statistic because it's a Wald, it's a distributed according to T distribution because it's a ratio of the beta estimate and the standard error of the beta from the linear regression. In this case, the regression has just one genotype vector as predictor and the phenotype as outcome. And it's the same as the significance of the Pearson correlation between the, so if we go back to the first slide, there is an outcome variable Y and a given feature for a given SNP, yep. Right? So the significance of the correlation between those two is the Z vector. So it's if there is no signal, it's distributed as a standard normal distribution, because under null z statistics is like a standard normal with unit variance.
And it appears that those z values are related with beta by design matrix A, right?
Yes.
And A matrix is squared matrix, and it's sparse banded matrix.
Yes, it is in its structure. It is basically this matrix, except this was a correlation among features. But now, because we are dealing with z-scores, we need to take into account the sample size and also the variance of each genotype vector. But as shown here, this A matrix is basically a rescaled correlation matrix.
So does it mean that we can find beta now by applying least squares?
Yes, in principle, theoretically, yes, but that doesn't work same as it didn't work with this OLS from the original data. And it's because there is too many, each one has too few effects, there is too much correlations. And I have one of the last slides about the different regularization strategies, because it's possible to make this work. Of course, either by like a ridge regression or by transforming into the principal components of this A matrix, kind of selecting the linear combinations of the SNPs that are, anyway, it's possible to make it work, but not in a kind of naive OLS way, not
okay.
And a typical, and also because, so now we are turning to these prior assumptions on the beta. It's a very common assumption that beta is sparse. Therefore, if we solve it with a regression model, it needs to be this sparse prior on the solution. We need to impose a sparse prior to force a lot of the betas to go to zero and only a subset of them to actually be significant. Because if we don't impose this assumption, then like there are no traits as far as we can find so far in human beings where every single SNP has an effect. It really looks like a fairly sparse distribution, and that's why it's closely related to a regression with a sparse prior. So in what we call a mixer prior distribution, it's just spike and slab distribution for the regression coefficients beta, which only a small fraction of them, by one, maybe 1% or so, have non-zero value described as a normal prior, and the rest are Dirac delta, right, equal to zero.
Can I ask a question, please?
Yes.
So I want to ask, is there any reason that you use this mixture model instead of something different for sparsity, for example, lasso regression, which has this Laplace prior, which is different?
I have it as a one of the, like, on a discussion slide. This is something we should try. We are looking for different priors that are more convenient mathematically, because this one is not the most convenient. So I think I saw papers where it's another priors being used, but surprisingly in our domain, kind of this prior is very much used. So it's used in so many publications that it kind of becomes a very conventional thing now. But there are other, like, continuous shrinkage priors, and I'm afraid of saying wrong thing, but horseshoe prior maybe. I saw other priors as well. The main thing, it needs to have a heavy tail. It needs to be more heavy tailored than a normal distribution. So I think the prior used in lasso would also work well.
It also seems to me that the great advantage of exactly this prior is the possibility to control the number of zero weights, this P1 parameter.
Yes. Yep.
Probably this is beneficial.
Yes. In the prior you mentioned, is there also a way to control, you know, the sparsity?
I'm not sure. Probably you just
Because
you can vary the dispersion, the variance of Laplace, but I'm not sure if there is a direct connection between the share of weights and the.
Because I think that would be, let's say, Cauchy distribution is like super heavy-tailed, but because you can't really control the kurtosis, let's say, right? So variance is just relates to scale, right? We sometimes just need to rescale, and that's just a matter of choosing units. But we actually need to control the kurtosis, and the pi1 in this mixture would directly translates to how heavy are the tails. So I think it's maybe part of the reason. I haven't been able to find another distribution where you can control this easily.
Okay. Thank you.
Okay, I think this is where I can give the word to Bayram, because this is just the same illustration of like there is these betas and these scores, they may correlate with multiple betas. So there are each SNP having beta would of course lead to a significant z-score for that SNP, right? But let's say, okay, the one I'm showing right now, it's a highest z-score in GWAS because it's associated with two SNPs, both having some correlation. This is kind of, I don't know if it's, and the idea of finemapping is to actually find those underlying betas, find those betas that are coming from non-null branch of our spike and slab distribution. Okay, Bayram, are you ready to take over? I stop sharing and you can.
Let me try. Can you see?
Yes.
Okay, so hello everyone again. Today I'm talking about extension of the MiXeR model, FINEMAP-MiXeR. It's a variational Bayesian approach for genetic fine mapping. And firstly, let me mention about what FINEMAP means in genomic literature. So as Alex mentioned, like we may have some trait of interest and we have already genome data. So we do some associations, literally called genome-wide associations, using some tools. The Plink is one of the most famous and most used tools to do such kind of association. It's fairly basic operation, but it requires some computation. So in the end, we got some summary statistics, like Alex mentioned, like z-scores or LD matrices that we can share with the other researchers or other people. So one of our other data that we can share is the Manhattan plot, which corresponds to the p-values of the whole chromosome here. And as you see, there are some peak hits around like chromosome 20, chromosome 19, or like 12, and so on. So these means that those SNPs are those SNPs has lower p-value, meaning that they have some stronger association in terms of statistics. But the question is, those SNPs with lower p-value are really the causal ones? And the quick answer is no, because there are some reasons to easily say this no. The firstly, because since we are like genotyping, the actual causal one could not be measured or imputed at this given study. So it's not, the causal one may not be there, and these SNPs can have low p-value due to the LD of the actual one. So this can be one of the cases. And the other one is actual causal one can be still there, but it cannot be detected. It cannot has the lowest p-value due to the insufficient power. So if we don't have enough data, if you don't have enough case control balance, or if the data is not well organized, so the actual causal may not be also detected in a given simple genome-wide association study. And also, like, even if it's there, like due to linkage disequilibrium, it can be lead to have a highly correlated association result and many false significant SNPs at the locus. So here, for instance, if you have a look to this table, assuming that this is some focused version of this Manhattan plot, and assuming that the red ones are the actual causal ones, you can see that there are some SNPs who has higher hits compared to the actual causal ones, and this is because they can be correlated with either SNP2 or SNP1 or both of them. So this may accumulate each other and make a higher lower p-value or higher hits compared to the actual ones. So fine mapping studies aims to like selecting the actual causal ones using different approaches to prioritize that this is the actual causal ones, and these are, they aim to eliminate the SNPs which has LD structure or correlation with the actual one. And there are many different approaches in literature, like using the heuristic methods or like some penalized regression methods or Bayesian methods. So if you are interested on this topic, there's a really good survey about that. Although it's a bit outdated, it's still like good to use to have an introduction and get the state of art. But what I can summarize here is among those methods, like Bayesian methods are the more successful and widely used in nowadays, because like they are more flexible and suitable to model the fine mapping issue, because like here we would like to model some sparsity, and these Bayesian methods are more successful compared to the heuristic ones or the regression methods. I'm not going to go into all of the methods available in literature, but I'm going to give a really brief overview. So there are some Bayesian methods available. So they are listed here. So these are based on the calculation of the posterior probabilities based on the given model. But one issue is assuming that we have m variants and k number of causal SNPs, we have m choose k possible combinations. So all these methods also relies on calculation these m choose k posteriors, and then have a search to choose the most possible one. And it's not so computational friendly. And now I have two other methods which deserve mentioning more. The first one is FINEMAP, tries to reduce the computational issue that I mentioned now. So they use some Cholesky decomposition methods to reduce the computation for calculating the posterior likelihood functions, which has some matrices on the which has covariance matrices that is on the order of m, which is so m could be great, and the calculation of the likelihoods would be on the order of m cubed. So they are trying to reduce it using some decomposition techniques. And also for searching, they use shotgun stochastic search. So due to this approach, they have fairly faster computation compared to previous methods by keeping the accuracy. So this is the basic selling point of this method. So it keeps the accuracy same as the previous ones, but much faster. And the other one, which Alex also is going to touch about the mathematical details, so I'm not going to mention so much in detail, but it's also now this one uses the Bayesian approach by optimizing the sum of single effects model, and it also uses the variational Bayesian concept here, and it gives in the end some credible set of being causal SNPs. And it claims better accuracy than all other methods in their paper. So in our approach, we are going to do the comparison with these two methods to advertise our method. And yeah, what is our method? So we are going to use the MiXeR model and variational Bayesian approach to obtain a FINEMAP method here. And it will be based on the optimization of the likelihoods based on the MiXeR model. So here we don't need any exhaustive search as other methods did. So we just need to go into do some optimization. And in the end, like we need the z-scores, we need the LD matrix, and then we calculate the probability of being causal of each SNP at a given locus, at a given region of the human genome. Okay, so like now some math. Alex already mentioned about the first part, but let me just refresh. So like phenotype can be modeled as a linear combination of the genome vector and corresponding effect size with an environmental factor here. And also the z-scores can also be modeled with the correlation matrix of the G matrix times the effect size. plus some error term here. So these are not equal. This is the environmental factor. This is the error term. And we model error term as a Gaussian distribution with a variance R0 square. So R0 square can be either assumed as a fixed number or can be predetermined or calculated. So it's not so big deal. Then we postulate the effect size of the SNP as a Gaussian mixture. So the first term corresponds to null terms, and the second term corresponds to the causal terms here. So Alex represent this as a Dirac delta here. So here we are also assuming that this is Dirac delta, but computationally we are using we use it as a Gaussian distribution with zero mean and sigma variance. So sigma is a very fairly low number here that we are going to use, but it's practically it's the same. So okay, now the distribution of the z-scores here, if we are using this this equation, we already know that error term is a Gaussian distribution. So if you know the effect size, the beta I here, and we already know the A matrix, so we can obtain the distribution of z-scores as a Gaussian distribution, whose means the linear combination of the A vector and beta I. And assuming that we also know the hyperparameters P1, sigma square, beta, and sigma 0 square, which can be either optimized by the Alex mixture model, mixture approach itself, or can be with can also be like solved by other approach. Assuming that we also know this, and let's define a latent variable UI. So here UI corresponds. UI is a vector that shows if it's 1. It's causal, otherwise 0, because in the end we would like to say that this SNP is causal with this probability, or this SNP is not causal with this probability. So we need to have another latent variable here. So we define a latent variable called UI and say that if UI is 0, the distribution of the beta I is the same as here, right? Nothing surprised that it's a Dirac delta distribution, zero distribution. Or if the latent variable is 1, we can say that the distribution of beta I is a Gaussian distribution with sigma square beta. So and we can also like model the latent variable distribution as a Bernoulli distribution, which is like which is not so surprising, I believe. So these are the model that we have started, and we can combine all these beta and nu and z using chain rule to have the full probabilistic model here. And then if we integrate with respect to u and beta, we can have the likelihood function of z given the theta, and this is some static point of the other methods, as Alex is going to present as well. So all we need to do is the optimizing this function here. And in FINEMApping method, my aim is to find what is betas and what is UIs here. So this is my aim to do in the following slides. Okay. So as I mentioned, like Alex also is going to present some other methods to solve, for instance, how to solve theta, I believe, or some other things. But here, my main aim is to trying to find beta and u here, and we don't know their distribution. Actually, we don't know the distribution of beta or u given the parameters available. So instead of that, instead of optimization this, we are going to optimize its evidence lower bound. So assuming that we have a Q distribution, any Q distribution, we are free to write this expression. So instead of the logarithm of the likelihood function, we write it with a arbitrary distribution here. And since Kullback-Leibler divergence is always positive, we can ignore this part, and we have a bound, evidence lower bound as follows. You know much better than me, so I hope you don't get bored. So here the question is, if the distribution that we have chosen is close to the distribution of the actual one, which we don't know if they are close to each other. So this is close to 0, and we are going to have a psi approximation of the actual likelihood function that we are going to optimize. And like we have chosen the distribution as follows. So for UI, we assume that it's a Bernoulli distribution, and for beta I, we assume that it has a mean, and each variant has a mean at nu I, and its own variance sigma square I. So, and also we assume that each SNP here, each beta I are independent from each other. That's why we have a product here, which is not actually due to the LD structure, but here this is the, for the sake of this, for the following computation, this is a quite convenient function that we can use, and we observed that it worked. So we didn't return back to change our parametric family choice. So this is what we use. And when we are using this parametric function here, our evidence lower bound becomes like that. So we are going to optimize this function in the end. So as I'm going to mention in the following slide that we will be using Adam algorithm to optimize this function. So basically, as you know better than me, it requires the first derivative of this function. So we are going to calculate the first derivative with respect to all the parameters that we would like to solve. So these are the parameters that we would like to solve. So it's like it includes some heavy maths here that like it's not so easy to present, but I liked while I was working. This is the most fascinating part of my end to do in this work. I will say that like solving the derivatives of all parameters. So if you are curious about the details, you can click, you can have a look on Bioarchive to have a look. But maybe I can say that for some of these special treatments, for instance, like we have used reparameterization trick because we have such things like the derivative of expectation of QA, derivative of expectation of QA, expectation of FP with respect to QA, the derivative of it with respect to theta I. It's harder to say, but easy to just points. Like it's harder to obtain such derivatives, and we have these on T1. And so in order to solve these things, like the reparameterization trick is widely used as an approach to such a computation. So we have defined some parametric families here as a function of the variables. So beta here we assume that beta is equal to nu times nu plus sigma times the parametric family that we have chosen, and then our expectations here for the first time becomes like that as a function of the epsilons. So this makes the calculations quite easier to obtain the derivatives. And like when we do the math after a couple of maybe 10 pages, then we obtain the derivatives with respect to all parameters of interest here, so that we are ready to do the optimization. And like as I said before, like there are some also hyperparameters that needs to be optimized or determined, like sigma 0 square, pi 1, or r square. So I'm not going to this not so big deal, because it can either be like estimated or calculated with other methods. So I haven't mentioned it. For instance, we also have the derivatives with respect to theta, which corresponds to these three terms. So this can also be optimized in our method, or Oleksandr tool, for instance, or some other tools available in literature. So I'm not going to the optimization of these tools in detail, but this can be somehow solved. And the optimization is done by Adam algorithm. So I don't want to like go into study, because you probably well know you probably know quite good. But I would say that since our variables like Qi or sigma I has some constraints, like Qi corresponds to probability of being causal, so it should be between 0 and 1, or sigma square R is a variance, so it should be between, it shouldn't be negative, for instance. So we have such constraint on the decision variables. So we would like to do some modification on Adam algorithm to run it. So first we are using reparameterization here, not reparameterization trick, but reparameterization. So instead of Qi itself, we are going to define Qi as a function of OI here. So we are going to optimize OI instead of Qi. So OI can be any number from -infinity to +infinity, while we keep that, we make sure that here, thanks to this function, Qi is from between 0 and 1. So then it's also quite easy to calculate the derivative of OI by the chain rule. So it's not so big deal to do such a change and then do a convenient optimization for Qi, for instance. And like we have calculated our derivative. We have applied the algorithm to optimize, and we got some results. For each SNP, we calculated the probability of being causal or the effect size of this SNP, such that. But then we would like to do some comparison, like how good our method works compared to the others. So we have done different simulations for that. Firstly, we evaluated the performance on synthetic data and then real data using the UK Biobank data. And we compare our method with the most successful ones in the literature, FINEMAP 1.4. So this is the extension of FINEMAP and SuSiE. So firstly, we do some simulations on synthetic data. So in synthetic data, we have created this data, so it's like fairly, we can share also with you. So it's not belong to human. So we have created a synthetic data, and we randomly choose a region whose length is M on this synthetic data, and we also assume that, we also assign that there are some number of causes in this region, 1 to 10. And we have also examined this for different heritability. Heritability means how the signal is good. Like the higher heritability makes it easier to detect the causal SNPs, for instance. So it's you can think heritability as a quality of a signal in that sense. And then, since we have assigned the causal SNPs, we have defined the heritability, we have the synthetic one. Then we can also assign a hypothetical phenotype. So here in this scenario, we are like a gut to create the genotype, define the causals, and then get the phenotype data. And now we have the all data. We have the G matrix, we have the Y, and then when we have the G matrix and Y, namely the phenotype and genotype, we can easily apply it to the GWAS to get the Z-scores and the LD matrix, because this is just a correlation of the G. And then using this as an input to our method and the FINEMAP 1.4 to get the actual posterior probabilities of being causal. And with the same for SuSiE, but SuSiE requires the actual data itself, because it's like, this is by the definition of the method that they are using the G and Y, and this is not preferred because G and Y is not always available, but then they updated their method to the summary statistics. But here we are using this because the performance-wise, they are not so different. And then we got the Q vectors, posterior vectors here. And then in order to do the evaluation, we calculated the true positive rate based on different false positive rate thresholds here. And then we do this until for all numbers from 0 to 1 to draw the ROC curves here. And as you see, the blue ones correspond to our method here, while the red ones correspond to FINEMAP, and the yellow ones is SuSiE. And as you see, our method performs better for different scenarios here, but it's like These are fairly limited scenarios that I can present here. And we also calculate the area under curve, so we can guarantee that our method performs better when we calculate the area under these ROC curves. But we have also like conducted this experiment for 31 different scenarios, differing from number of causals, number of SNFs, or heritability. So we did for the extensive simulations, and like the 65% of these scenarios, I would say that our method outperform compared to the others. So these are the cases that the other methods outperform us. Now the other scenario is simulation with real data, and it's a bit challenging compared to the actual simulation data, because here firstly we are using UKB genome data, and height is chosen as a phenomic data here. So firstly, in order to do an inference on this data, we would like to eliminate the effects of some covariance, because when we are talking about height data, the height of the man is expected to be taller than height of the woman. This is as an expected ratio, so as expected value. So we would like to eliminate such things like the age, sex, or there are also some other things in order to get the purely genomic effects. So firstly, we would like to eliminate the effects of those. And once we did such elimination, then we focus on some heritable loci on the genome data, because there are already some GWAS studies available. So we use these studies to find the heritable regions. But the problem here is that previously we know the ground truth data, which one is causal, which one, but here we don't know it. We don't know which one is actually causal. We just have the how do you call it? We just have the GWAS studies. So we just have some numbers some associations, but we don't know the ground truth. So instead of like having the area under curve or rock here, we are trying to use estimate the height phenotype using our FINEMAP and FINEMAP results, so that we are trying to get some R-square to make it to do the comparison. So this figure like summarizes what I want to say here. So we have the data, genome matrix and Y, and some covariates that we eliminated from the phenotype. Then we divide the data into two. One can be considered as train, the other can be considered as test. So let's focus on train here. So we eliminate the effects of covariates here, and then we applied some again GWAS as we did before, and then we use these as an input of the methods, and then we get the posteriors. Once we get the posteriors, we are going to use these posteriors to estimate the phenotype using the multiple linear regression, and then we get some weight, some effect size to estimate the phenotype here. And for test data, we also use this V vector, W vector, to estimate the test data, and then actually we obtain the R-square test here to do the comparison. And as I said, like we estimate the W vector with 80% trained data, and then calculate the R-square test for 20% data. So we did this for 15 different cases. And yeah, these are the sum of them here that I present. So the R-squares are quite small. So this may look suspicious, but actually it's not, because height is highly polygenic. So there are locus everywhere that is heritable when we consider height. So even if we are focused on the most heritable loci, their effects are limited, I would say. So it doesn't seem so big as you may expect, but in the end, if you compare with the other locus, it's fairly quite big. So in these 15 cases, we observed that in 11 cases, we have the better results compared to others. So like this is also a good aspect. And yeah, I think I have a couple of minutes. So also the other aspects of the FINEMAP method are computation complexity. So For instance, SuSiE has k times n order of computation. So here k corresponds to number of causes, m corresponds to size of the locus, number of SNFs, and n corresponds to number of samples. And for SuSiE RSS is k times m squared, and for FINEMAP is k squared m. And in our method relies on the calculation of the derivatives in each Adam algorithm. And if we write down derivatives in a compact form, the second and third one is not problematic, but the first one, this term, a squared times u, is the matrix vector multiplication, and it requires m squared operation here. So if we somehow solve this with a lower computation, we can reduce our computation requirement. But for now, naively, it's order of m squared. But on the other hand, we are able to compute this using some principal component analysis approximations here, so that we can calculate this in a lower dimension and then project again. So this reduces the computation of this importantly reduced, like PC times m, where PC is the first PC component of the matrix of interest, covariance matrix of interest. So it's fairly reduced the computation. And these are the some comparisons. So first row corresponds, what if we change the number of number of SNPs that we are going to examine. So the first column corresponds to us, the second column corresponds to SuSiE, and the third column corresponds to FINEMAP. So as you see, like actually our first naive method is computation increase with quadratically with as m increases, but in our other PCA method is almost linearly increases. So this is a good aspect. While here the SuSiE RSS, for instance, also increases with the m square. So in that sense, our naive method, the PCA method, is comparable with the FINEMAP method, which is also linearly increased. But on the other hand, for instance, if we increase the heritability, our method doesn't change, while SuSiE as well doesn't change. On the other hand, FINEMAP increases because of its shotgun stochastic search requires some more time to evaluate all possible configurations. On the other hand, if we increase the number of causes, k, here our method stays constant, while SuSiE, both SuSiE and FINEMAP increases. So if we consider the whole picture here, that we can say that our method linearly increases m if we apply the PCA approach here, while it stays constant with k and heritability, number of causes and heritability. On the other hand, the other methods either increase with k, number of causes, or heritability. So in that aspect, our method is more stable in terms of computational complexity. So to sum up, we have proposed the FINEMAP method that can outperform other methods in the literature in different aspects that I showed you now. And it can be used to FINEMAP a locus, or also it can be used to estimate the phenotypes, although there are some other estimation methods. These methods can also be used to estimate the phenotype. And we have already submitted the preprint that you can have a look about the details, and it has been also submitted to Nature Communications. And one of the next steps is using annotations from GSA-MiXeR that Alex is going to present you now, I think briefly, so that to prioritize some SNFs, because for now we assume that we all have the equal priors at the beginning. So if we inform from Alex method GSA-MiXeR, we can use these priors and improve the performance further. And yeah, these are the some references that I want to put here, and I would like to thank you for listening and also your contributions, Dimitris. I know that you had a really good contribution in this work.
Well, Bayram, I have one question.
Yeah.
It seems that in your model, in your joint distribution, it is possible to integrate out with respect to beta. So if we fixed U, we can integrate out our beta, like analytically. Yeah, in this joint distribution. And then in this case, we need only to model Q distribution only on U term.
Yeah.
Have you tried this way of inference?
You are right, but actually our main aim is to find U.
Well, yes, but okay. And
I have a slide on that. We didn't integrate it in the like elbow optimization, but like throughout the process, I realized that this is the case, and we used it elsewhere. So I have a slide. When if I share that slide, maybe we can see how that runs. So I think we haven't tried in this particular application, and I do see that that's a room for improvement.
Yes, because now it seems like that you use like very simple factorial assumption on a Q function over beta. So beta is actually a multidimensional vector, but you approximate it with component-wise normal distribution. So if we integrate out beta, we don't need to model Q on beta, and we just need to focus on Q of U, and maybe it would lead to some better result in terms of Elbor.
And the question is, how are you going to model Q of U? Are you going to model it component-wise?
Well, in the same way like here, factorial distribution.
And what would be the benefits of integrating out beta then?
Maybe
sorry, once again, there's some problems.
What would be the benefit of integrating out beta if you still model Q of U using factorial structure?
Maybe it may reduce the required computation, but also maybe you may, I don't know.
But if you don't need a parametric form on a posterior
because we do not need to
beta,
model Q of beta.
So maybe this posterior as a normal with mu and sigma imprecise, then
So there'll be just acceleration, yep, in computation time, improvement of computation time, but not the improvement of performance.
There could be more accuracy gained by not imposing a specific parametric form for their, like in the Q function, if it's only a Q function of the U, but not beta, right? So we don't need to approximate the posterior of the beta through a normal distribution with mu and sigma I. Right, those can be factored out, and if it's not really a normal but some other distribution, that could save. It's 3 times less parameters. That's a computational aspect, but also the fact that we bypass altogether the need for posterior approximation on beta could be an interesting.
Yeah.
Okay.
Maybe we could also focus on more sophisticated distributions when we just need to estimate the UI. Yeah, we should have a look at Alex, I think.
Okay, I think we'll have an internal discussion with Dmitry, maybe even tonight. And if there's really some chance of improvement, we'll let you know. Okay.
Bayram, can I share the rest of my slides, because it's more or less one of the 2 things I wanted to show. Okay, so basically one was this a bit extra information on the SuSiE, or the sum of single effect model from the Stevens lab. This group that I put here on the title is actually very strong in kind of applying Bayesian methods to the statistical genetics, both Markov chain, Monte Carlo, variational bias, and like prior to like talking 3 years ago to you, I couldn't even follow what they do. I think now I'm a little bit started getting into the papers, still not very extensively, and they have some tools on GitHub. So this is just kind of a summary slide of what they have. But so SuSiE, if a method that also Bayram compared to, and it seems that our implementation outperforms theirs, is basically, if I can simple thing is to replace in the Q function, right? The approximation of the posterior on the latent parameters. If we replace our Bernoulli with the multinomial, then FINEMAP-MiXeR turns into SuSiE, right? So if they are modeling the effects as a sum of multiple betas, that's hence the name, the sum of single effect model. And the point of single effect model is that each beta is still a vector, right? So in the first formula, it's a sum of the vector betas, where each beta has just a single non-zero element, right? And the point is that now the probability, in like which one is having this non-zero effect, which component in the beta vector has a non-zero effect, is a multinomial distribution. So kind of instead of postulating that each variant has a posterior probability of being non-zero, they are allowing for a certain combination of variants to have together a probability of being non-zero, but they sort of allow this uncertainty about which variant is a causal. If you think of those cluster of highly correlated SNPs I shown on the LD matrix, right? Sometimes you just cannot pinpoint the exact causal variant, which one has non-zero beta, but you can say it's one of these three or one of these five, right? And I think this is like a more instead of factoring out the Q distribution across SNPs, they are allowing for a multinomial distribution. I don't know if that explanation was any good, but it's like they are posterior under single effect regression. So again, the posterior is a sum, so the beta is a sum of multiple components, and each component has a posterior very similar to our Q function, but now what was this latent U variable, instead of being a Bernoulli, it become a multinomial, and this alphas defining the posterior probability of different components in that vector to be non-zero. They are kind of allowing for certain structure of the posterior U distribution. Is it?
What is P here? Is it dimensionality of beta?
Sorry, pi or P? Yes, that's a number. This is like the simplest prior of all SNPs have equal prior probabilities. Yes, P is a number of the dimensionality of beta, correct?
Okay, thanks.
And then if we assume that there is just one non-zero beta, then instead of allowing like some posterior, so they allow yeah, anyway, I think this is like relaxing the assumption of factorization for the U latent variable, and it's kind of the underlying idea of their flagship SuSiE model. And then the inference of this model is, of course, a bit tricky, because you wouldn't like basically now if you have multinomial distribution in your posterior governed by P, right? The dimensionality, dimensionality of how many SNPs, dimensionality of beta, right? So, but then you also have a sum of them. So the total amount of parameters is now a product of like how many non-zero betas you allow times the number of variants we are modeling. Instead of, in our case, it was 3P basically, right? 3 times the number of variants, because we have U, mu, and sigma, right? Just 3 parameters per every variant, and for them it's gonna be a lot more. So I'm not sure if it's still possible to apply Adam optimization in this space. They had to do this kind of stochastic search, like more empirical, like searching for the subset of parameters which which they are optimizing. Anyway, so this was just my little insight into our difference between our model and SuSiE. So they are allowing for a more flexible model, for a more flexible Q distribution. And nevertheless, our model came out more accurate, probably because of direct elbow optimization with Adam, rather than some stochastic search procedure, which eventually was lost in the flexibility allowed by the underlying posterior distribution.
But didn't they optimize elbow as well in their model?
So I know that they definitely did optimize elbow in some of their other papers, let's say the first one from 2012. Whether, like, exactly what was the inference procedure used in their 2020 paper introducing SuSiE, it was a little bit hard. I didn't get to the bottom of that. But I think I'll continue. Bayram, do you remember?
Like, they have mentioned that their optimization is equivalent to elbow approximation. So they are using some.
I think it has basically more heuristics, more kind of just more empirical choices in the code. That's my, because overall they have a wider class of the posterior distributions that they can describe, and it's reasonable to represent some uncertainty about like not being able to choose exactly which SNPs have an effect, right? But just allow for sets. You have a subset of 3 or 5, and one of them have an effect. You cannot be more certain than this. Okay, but if so, anyway, SuSiE is something for us to learn from, and maybe see if we can like extend our model to also be flexible, more flexible in its definition of the U posterior. But Dmitry, what you suggested about integrating out from betas, this is like a reiteration of the slide I shown before, now with a proper mathematical notation, without elbow, right? Without approximating the likelihood, we've been using something I call the sampling approach, and I think now I can explain it a bit more straightforward. It was like, instead of Markov chain Monte Carlo sampling from posterior, we were just sampling the prior for the U distribution. Right, basically sampling without introducing the, like, how do we call it, Q, I think, right? So without basically just sampling from this prior Bernoulli distribution on the latent variables. If you do enough samples to approximate this U integral here in the middle, then the beta is just a normal distribution with an analytically derived variance shown here in the bottom, and it's all very tractable likelihood to maximize. So my understanding is that we can now kind of do a hybrid between this really stupid prior sampling for the U. For U, we can do the proper elbow optimization, but then we can benefit from this second integral being analytical. Right? Is this the idea?
Well, but it seems like if you sample from prior distribution, you do not account for probable U. So you should sample U like from posterior distribution, from those U that somehow correspond to Z, to the observed variable. If you just sample from a prior, most of the point would be inefficient.
Yeah, that's very inefficient. It's not biased, right? So this still gives an unbiased estimate for the theta parameter, but it's extremely inefficient in terms of compute resources. That's for sure. So we would not be able to do it for the Bayram's approach. We only did it when the space for theta was like 1, 2, 3 parameters, very small. If you're only interested in the estimating the prior by one parameter, just a single parameter in the model, this still worked if you do enough samples. Like the number of samples was 20,000 or something, maybe 100,000. But yeah. So absolutely, we will move away from sampling from this prior on U. But I think this is basically the hybrid we need to make. We can take the analytical shape for this second integral over beta yeah, and given u, right? That's analytical, and then
and then try to model Q of u in factorial setting, but it's some more complex assumption on distribution over u.
Yes.
To make it somehow correlated in u variables.
Yep. I think this is a very good conclusion and next steps for us to work out this kind of system. Take the best from SuSiE, take the best from what's possible analytically, and plug this into the elbow optimizations the way Bayram is doing right now.
I have one additional question. Is there any evidence to suppose that two neighboring genes are more likely to be relevant? Or is it usually just one relevant gene and all other correlated genes seem to be irrelevant?
I think there is definitely some localization within the genome, so at least within one gene, yes, and that's like on this slide, I have another model where this sigma parameter, it's not a single one. So the prior assumption here is that within a gene, there could be a higher or smaller variance of effects than on average. That's this GSA-MiXeR I briefly mentioned. So within certain localized regions, yes, whether it is like several adjacent genes or just one of them, it's sometimes it's like there are big regions like hotspots, like MHC for schizophrenia has many hundreds of genes and all seems to have something to bring to the phenotype. But sometimes
then I think that it could be really useful to use to apply non-factorial Q of u, and you might model it as, for example, Markov chain, because you have a definite relational neighborhood. So you understand that some genes are neighbors to each other, some genes are not. So you may model it as a Markov chain, first order or kth order Markov chain, and then you obtain a much more flexible approximation for a posterior view. So that's really worth thinking it over.
So I think, yes, also looking into the Markov chain here would be very good for us, because I felt people, if they apply, like, my concern with the current methods was that if they apply Markov chains, they apply it on everything, including beta, and then it gets pretty difficult to sample from, like, I didn't see the point if we can here do it analytically, to do a lot of samples from a normal distribution. This was a little strange, but if we can work out this hybrid solution where we do Markov chain samples for U, but not for beta, that could work, I think, very well.
But it seems that in this case, you would need a Gumbel softmax or similar tricks in order to optimize ALBA with respect to parameters of Q of u, when now Q of u is non-factorial, but more.
Well, yes, but that's what you have suggested. That immediately, since we have a non-factorial Q of u, we can no longer perform analytic integration with respect to Q of u, because it's no longer factorial, and we need to apply Gumbel softmax trick. But that's even more interesting, I think. More mathematics, more equations.
Could you point us to that Gumbel max, or what? Gumbel max. Yeah, you mentioned it last time. I think I still haven't get to the bottom of that paper.
Don't worry. In 2 weeks, I will read the lecture in Moscow University about Gumbel softmax tricks. So you may
if you can
upload it on YouTube.
Yes, then we'll. I see we are running out of time. Let me skip this one and skip this as well. So the discussion, just briefly, since it's an update since last time, we briefly touched on this, the first one, with other priors that are allowing for heavy tails, but kind of are reasonably tractable when we do the linear combinations. So I think still it's our prior is kind of has some advantages, and we keep using it. We learn how to do posterior. That's a new thing for us, and we can improve on that. Adam is just a brilliant thing. Previously, we used differential evolution, and then like Nedler made some zero-order methods, which can handle 3 parameters, maybe 5. And now we can do like many tens of thousands of parameters as in GSA-MiXeR. This is amazing. I'm extremely impressed by Adam. Simple, and yeah. I can also skip conclusions, the practical things. To return about the courses, our group is also teaching a course on the GWAS, and it's an open registration. I can share the slides, and there is this registration. It's a 4-day course, end of March, free, and it's on Zoom. And there are some exams, but you don't need to take the exams. It's possible to just listen. It's totally many students just get for the lectures. So it's my group, our group. It's, of course, is organized by my colleagues Alexei Shadrin. And if you're interested in this domain, if some of the students are interested in learning about statistical genetics and overall this GWAS, and psychiatric genetics, this is a good opportunity to kind of join those 4 days of GWAS course. It's probably the 4th or 5th time we are teaching it, so it kind of already more or less polished. With some from clinical perspective, some from mathematics, some from more like practical application. So yeah, there is a previous year program here as well, if you're interested. And then if you want some more hands-on things, I have a little like GWAS 101 with some Jupyter notebook if you want to run some code and maybe put your hands on some of this data, some artificial, some real. So that's also another link. And another introductory video, if you want more of my first introduction, I think 20 minutes we spent in the beginning. We did it also 2019 with my colleague Kevin O'Connell at the MIPT seminar. So there is another link here. So yep, that's just some useful links. Otherwise, the conclusions from our part is we are very excited about statistical genetics. It's a very large and active research area, many promising applications of Bayesian inference and other things. Challenges are this very high number, like millions, sometimes tens of millions of genetic features with tiny outcome and a high correlation. So even this zero-order, simplistic, embarrassingly simple model with linear combination gives a rise to lots of pretty interesting math. And then we presented FINEMAP-MiXeR, which outperforms some of the existing algorithms, and it's all based on ELBO and Adam optimization. And we hope that some of this can actually lead to our better understanding of the complex psychiatric disorders, schizophrenia, and maybe even better treatment at some point. So we have a strong motivation for doing this. That's, I think, all Bayram, that's all we had, right?
Yes, I think so. Yeah.
So if there is more time, we can discuss.
Well, first of all, thanks, colleagues, Oleksandr, Bayram. Thanks for a very nice talk, and it's always a pleasure to see that Bayesian methods find more and more applications in different areas. So our time is almost up, but I think we have time for a couple of quick questions. So any questions, guys? Or I guess that maybe all questions have been asked during your talk. And I hope that, well, we've suggested to you at least a couple of ideas that are potentially fruitful. And I've written down some derivations. I think that Dmitry was right, that it's really possible to integrate out beta and to come up with more compact expressions, which potentially would allow you to use more flexible approximations for posterior. So this is at least worth considering. And that's good. I think that this was one of the targets of your talk, in order to come up with some new ideas.
Yep. Can we take a screen, like no screenshot, a photo, like just mobile photo of your equations?
You mean
the one you did right now? I'm afraid what you did in like back of the envelope computation will take us another year to reproduce on our own. We got the direction, but if you can send us,
they look like this.
Yes, that's what we want.
They're not formatted. So I think it's better if I write it in a clean manner, because I was doing it was just for myself.
If you can send us some of these pointers and also the link to the lecture, so I would be very happy to join and
well, please remind me in 2 weeks. But
Okay.
maybe even in 2.5 weeks, when it's already available on YouTube, and then we'll send the link.
Thanks.
And about the equations, I think that yes, me or Dmitry, maybe we'll together, we'll write down some equations and we'll send it to you.
Thanks a lot. Thanks.
So good. Thank you, colleagues. Let's keep in touch.
Yes. Goodbye.
Thank
So
you.
cheers.
Thanks.
Bye.